Okay, everybody, welcome to the blood lab. This is follow up to the lecture material that we had during the lab period last time we were together. And what we're going to talk about today are the characteristics of blood, why blood is important, and how to recognize the different components of blood and what they do. So, for the purposes of Chapter 10 for the lab practical, you're going to need to understand um, what blood is composed of. You're going to be able to describe the process of hemostasis, and you want to be able to understand the results of a blood typing test. Make sure that you um, know how and where the blood cells are made, understand hemostasis. Um, for Chapter 12, that we're going to talk about body defenses, both specific and non-specific. You're going to want to understand the difference between nonspecific and specific defenses. You're going to want to understand where the tonsils, the thymus, the spleen, and the Peyer's patches are located. Um, and you want to be able to have basically a broad idea of how these different types of systems work. So blood is one of two fluid tissues in the human body, the other being lymph, and it's a connective tissue because it has very few cells and a lot of extracellular matrix. The components of blood include the living cells, which we call the formed elements, and the non-living component, which we call the plasma. One of the things that we can do with the formed elements of blood is that we can break them down into several broad classifications. Um, three very broad classifications include erythrocytes, leukocytes, and thrombocytes. The erythrocytes are the red blood cells, and they are primarily responsible for carrying oxygen and carbon dioxide around the body, as well as maintaining a relatively constant pH in the blood. The leukocytes are the white blood cells, and they're our body's defenses. What they're capable of doing is defending you against components called pathogens, which can either be living or non-living um, entities that are capable of making you sick. Um, the thrombocytes are the platelets, and they're involved primarily with blood clotting. If we were to look at the leukocytes, we could break them down into two broad categories: the granulocytes, which contain cytoplasmic granules that stain when they're treated with a particular type of dye in a blood smear, and the agranulocytes that do not have granules that stain with this same type of dye. If we were to categorize the white blood cells in order from most to least abundant, we would find that the neutrophils are the most abundant in a typical blood smear, followed by the lymphocytes, then the monocytes, the eosinophils, and the basophils. Now, of these, we should note that the granulocytes include the neutrophils, the eosinophils, and the basophils. The agranulocytes include the lymphocytes and the monocytes. Okay, so those are the agranulocytes. And there's a way to remember this, which is the mnemonic shown here: never let monkeys eat bananas. Okay, now, will these abundances change? Yes, they can, depending on whether or not you're battling an infection. You may see a difference in the percentage makeup of these white blood cells in a smear taken from somebody who might be perhaps fighting a particular type of an infection. And we'll talk a little bit more in detail about the individual role that each of these play in keeping us disease free. Now, this is a photomicrograph of an actual blood smear, and what you're seeing here are really a pretty fair representation of the formed elements of blood. Let's start with the tiniest formed elements. Those are the platelets, also known as the thrombocytes. You can see them here. They stain sort of purple with this dye that we use to treat blood smears in order to visualize the formed elements. They do not have a nucleus, and they contain large amounts of proteins called clotting factors, and this helps when we're injured to keep us from bleeding to death. You would mistake them almost for dirt if you didn't know that in fact those were platelets. The erythrocytes are these round, they look like little brown rings almost. They're actually biconcave discs. They're filled primarily with hemoglobin, and their job is to carry oxygen and carbon dioxide around the body. They also lack a nucleus because they are designed to maximize the inside of the cell to do nothing really but carry these two gases around the body, and so we didn't want to take up extra room inside the erythrocyte with cellular organelles that really aren't terribly important to the function of a red blood cell. You'll also notice that the erythrocytes tend to be pale in the middle, and they are a little bit darker around the edges. That's because the rims of the erythrocytes form sort of a ridge. Okay, they look like little biconcave discs. If you were to look at them head on, okay, they would have this kind of a shape to them. Yeah, that's not very good. Let me draw that again. Look a little bit like this. Like that. Where these will be the edges. Okay. And this will be the center. And the reason for that shape is to maximize area available for gas exchange. So those are the erythrocytes and the thrombocytes. Now, if we look at the formed elements with regard to the leukocytes, the white blood cells, here we're examining our disease fighters, and you can see some of the formed elements here. You can see neutrophils. Let's circle the neutrophils here. Okay, there's one, and there's one. The neutrophils have an unusual looking nucleus. It seems to have several lobes, and if you look very closely you can see pink staining granules in the cytoplasm. This is the most abundant of the leukocytes, and it's also the most abundant granulocyte. They are primarily designed to fight bacterial infection. They engage in phagocytosis of bacteria, and they also initiate chemical attack on infected cells. You can also see a lymphocyte in this picture, and we'll circle that here. Okay, and that's our lymphocyte. And what lymphocytes do is they provide our specific defenses. Essentially, the way a lymphocyte deals with an infection is that you have an army of about 1 billion different flavors of lymphocytes, all which can recognize a different antigen, which is essentially a molecule on the surface of a cell. And from among those 1 billion different types of lymphocytes, when we have a pathogen attempting to produce an infection inside the body, what happens is that we select for those lymphocytes that are best able to bind to the antigen that's on the surface of the invading pathogen, and then we stimulate those lymphocytes to divide and generate an army that can go out and specifically attack and destroy the invading pathogen. Lymphocytes come in two broad categories. There are the B lymphocytes, which mature in the bone marrow, and the T lymphocytes, which occur in the thymus, which is a lymphatic organ that's located in front of the heart and behind the sternum. The B lymphocytes produce antibodies, which are magic bullets that are capable of binding to the pathogen regardless of where the antibody is produced, because eventually it'll drift through the bloodstream and find its intended target, just like a heat-seeking missile does. And that's what we call our humoral immunity. The B lymphocytes do that. And the T lymphocytes are responsible for what we call our cell-mediated immunity. They actually have to show up where infected tissue is located, and then they initiate a chemical attack that will compromise the membrane integrity of the pathogen or the infected tissue, and cause the cytoplasm to leak out and the cell to die, or they will degranulate and initiate a sequence of events that causes the infected cell or the pathogen to commit suicide. So those are our T lymphocytes that are responsible for cell-mediated immunity. Now what you're going to want to be able to do when you look at the smears that are in this presentation and also in your textbook and also under the microscope in the lab is to identify the appearance of the different formed elements based on their color and based on their abundance, and then be able to describe what it does. Okay, so to help you out, what we have here is table 10.2 from your text that gives you a rundown of each of the different types of formed elements, the abundance of them in a particular volume of blood, a cubic millimeter, a description of their appearance, and then a description of what they do. Okay, so my advice to you is to very carefully study the material that's in table 10.2 and be able to recognize this if you see it on a PowerPoint or on a microscope slide. Okay, so that's 10.2. You can see here the different types of cells. Okay, so this is an actual blood smear. This is some practice. What you can see here, of course, are the erythrocytes. Right, those are very easy to pick out, the red blood cells. The lymphocytes are going to have these very large oval to round nucleus and very little cytoplasm. You can see here the neutrophils, okay, which have these unusual looking purple staining nuclei with many different lobes and these pink staining granules. And you can see here a monocyte. There's a monocyte and there is a monocyte, and what they eventually become are macrophages, which are capable of phagocytizing pathogens, chopping them into little pieces, and then displaying them to the immune system, which will become aware of their presence and attack. And then the eosinophil, you can see the red staining granules in the cytoplasm, and also a many-lobed nucleus. These are responsible primarily for fighting parasitic infections, and they also mediate the allergic response. And then down here you can see the basophil, which has also a many-lobed nucleus, but it's obscured by all of these purple staining granules, which contain a very potent inflammatory compound called histamine. What the basophils do is they promote the inflammatory response in the face of an infection, and the purpose of the inflammatory response is to bring blood to the area so that we can begin the process of destroying the pathogen and then eventually send out chemical signals that will bring cells into the area to repair the damaged tissue. So you can see all the different cell types here just contained in this one little smear. Okay, so again, lymphocyte, monocyte, eosinophil, neutrophil, basophil. Okay, another lymphocyte down there. The only thing you can't really see that well in this smear are the platelets, and again, they're extremely small and they lack organelles. But we've got some other examples of smears where you'll be able to pick these up relatively easily. Now, how is it that blood cells are made? Well, in the red bone marrow, the hemocytoblasts produce other stem cells, which then produce other lineages that eventually generate all the lymphocytes and then all the rest of the formed elements. So we start here with the hemocytoplast in the red bone marrow, and then depending on where the cell migrates, it will be exposed to chemical factors in the environment that cause it to produce what we call a myeloid stem cell, which is going to be fundamentally different from the hemocytoblast and will generate all of the granulocytes in addition to the monocytes, platelets, and erythrocytes. Or they will be basically escorted to a compartment where they're on counterchemical factors that cause them to move down the lymphoid line where they will produce the B and the T lymphocytes. Okay, so these are agranulocytes, remember, as are the monocytes. These are our granulocytes, the neutrophils, the eosinophils, and the basophils. Now you might ask, okay, where is this magical red bone marrow? And it's found in several places. It's found in the ribs and in the hips, in the clavicle and the scapula. It's found in spongy bone. It's found in the heads of the femur. In the developing skeleton, it's also found in the long bones, but in the adult skeleton, we only find red marrow again in the proximal heads of the femur. So this is where our blood is continually made, essentially in the areas that surround the bony thorax. Now you can see here, this is another example of a blood smear. You can pick out the neutrophils here and here. You can see the platelets, okay? Those are platelets. You can see the erythrocytes scattered throughout, okay? And here you can see a lymphocyte, right? You can see this large round to oval nucleus in the small amount of cytoplasm, which identifies it primarily as the agranulocyte known as the lymphocyte. Here you can see an example of neutrophils, lymphocytes, erythrocytes, and platelets. Okay, there you can see a neutrophil. There's another neutrophil. You can see a lymphocyte down there. Of course, all these little, they look like pink circles. Those are the erythrocytes. And the platelets, you can see a platelet there, and you can see a platelet there, and there, and there. Okay, those are the thrombocytes. This is just another close-up. Here you can see an eosinophil. Notice the red granules and the multi-lobed nucleus. These, of course, are erythrocytes. There's a platelet, okay? And over here, you can see the eosinophil lower magnification. You can see these erythrocytes here, and you can see a platelet here. Here you can see erythrocytes. You can see neutrophils here and here. There's a lymphocyte, okay? You can see some platelets here and here. You can see a platelet there and there, okay? So basically practice looking at these blood smears so that you get very used to what these different formed elements look like. Here you can see basophils. Notice the purple granules that almost completely obscure the nucleus, and notice the erythrocytes all around here, okay? Again, erythrocytes through here. There's the basophil, all right? You can see a platelet there, okay? I think you can see, if we look very carefully, I don't see any platelets in this particular photograph. Over here, okay, you can see a monocyte. Notice that there's more cytoplasm in the monocyte than there is in the lymphocyte. So that's the way to tell the two agranulocytes apart. Notice also that the nucleus has a kidney shape to it. And you can see here platelets. These are erythrocytes. Okay, there's a platelet, there's a platelet, there's a platelet and an erythrocyte. Here, erythrocyte, 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 monocyte right here. Okay, platelet there. Okay, another platelet, another platelet. And here you can see another blood smear, a lymphocyte, okay? There's a neutrophil, there's another neutrophil. You can see some platelets here and here and here. These are erythrocytes, okay? These are erythrocytes. So get used to looking at these blood smears because they're not all going to stain the exact same colors, but you can look for the general shape of the nucleus as a major clue, and also, again, the presence or absence of those granules in the leukocytes as a way to tell you which formed element is which in relation to the leukocyte population. Now, hemostasis is the process whereby we avoid the excessive loss of blood in the face of an injury. So if we look at the process of hemostasis, it starts with tissue injury. It could be a puncture, it could be a laceration, and what will happen is you'll begin to bleed. As this occurs, the damaged tissue will produce chemicals that will cause the smooth muscle in the middle layer of the blood vessel to contract, and this is going to temporarily stem the flow of blood out of the wound. Okay, this is what we call vascular spasm. Next thing that happens is that due to the damaged tissue exposing collagen fibers, platelets will begin to stick to the collagen and form a temporary plug. As the platelets stick, what will happen is they will release clotting factors, which will eventually trigger the production of an insoluble protein called fibrin, which will trap formed elements that drift by and form a temporary seal around the area of the wound, which will provide time for white blood cells to come in and remove any potential pathogens and get rid of any dead cells, and then eventually fibroblasts will come in and repair the area of the wound, and there will also be factors that will promote the division of the endothelial cells, and eventually the entire blood vessel will completely heal. And at that point, the fibrin clot will dissolve and everything will go back basically to the way it was before the injury began. When we look at the chemical reaction that leads to coagulation, we have to be aware of the fact that there are components that are always present in the blood plasma that are going to be part of what we call the clotting cascade. And essentially what a cascade is a way to take a very small initial signal and amplify it so that it's very, very large. Okay. Essentially, when we start a cascade, what will happen is it will have some sort of an event that will trigger the activation of an inactive molecule to an active molecule. So we'll have the inactive molecule here indicated by just a little circle, okay, and the active molecule indicated here by a circle with a little slash next to it, okay. What will happen next is that activated molecule will activate another molecule, which we'll indicate here by a box, okay, and here a box with a slash next to it, and then this will activate another molecule, okay, indicate this with a diamond, okay, and so on. And eventually what will happen is it will get back to the last molecule in the cascade, and that will produce now the desired effect. The reason for taking so many steps is that at each step an activated molecule can activate more than one molecule downstream, and so at each step in the cascade the signal gets bigger and bigger and bigger geometrically until we get to our end point. The other nice thing, of course, about the cascade is that all we have to do to turn it off is to stop now the first step, and then the whole thing shuts off, just like cutting off the faucet that might be supplying water that's running down a waterfall. Okay, basically everything will stop at that point. So that's the utility of a cascade. And in the blood, we always have an ample amount of calcium and quite a few clotting factors that are just in the plasma. Okay, so these are always around in the blood plasma. What happens when we have tissue injury is that we release chemical called tissue factor, and also as the platelets adhere and rupture, they release platelet factor, and that, along with the rest of these components in the cascade, will eventually result now in the production of a fibrin clot. Okay, so it's this series of events here that I'm trying to illustrate here that's happening with this big blue arrow. Okay, we're taking a very small initial stimulus, and we're blowing it up really, really big. The last few steps in the cascade is the production of a molecule called thrombin, which converts a soluble plasma protein called fibrinogen into an insoluble protein called fibrin. And the fibrin is what ends up trapping the formed elements and forming the temporary seal between the inside and the outside of the blood vessel. And this allows time for tissue repair to happen as the endothelial cells that die are removed and as the living ones are stimulated to divide, as fibroblasts come in and repair the damaged tissue. Eventually what happens is that the fibrin clot is no longer needed, it's destroyed, and then the injury has now been repaired. If we're missing any of these components, if clotting factor, if we're missing calcium, what will happen is that the clotting will not occur. And there are individuals that are born with a hereditary lack of some of these clotting factors, and they're called hemophiliacs. And what happens to them when they're injured is that they bleed continuously, they're unable to affect hemostasis, and the result is massive blood loss. The fix for this is to provide the missing clotting factor as an oral or as an injectable so that they're capable now of essentially generating a clot and allowing the injury a chance to heal. Most of these conditions are more common in men than in women because most of the genes for hemophilia lie on the X chromosome. Since a man only has one X chromosome and a woman has two, and since most of the hemophiliac conditions are what we call recessive conditions, where you need to have two bad copies of the gene for the disease to show up. In a female, you'd have to have a bad copy on each X chromosome, whereas in the male, if he has one bad copy, the Y chromosome has no genetic information to cover up for the defect in the bad gene on the X chromosome. So what happens is that the disease will appear, the disease will show up. And that's true of a lot of conditions that are on the X chromosome. They tend to be much more common in men than they are in women. Other examples of conditions that are on the X chromosome besides hemophilia include color blindness and male pattern baldness, just to name a few. Okay, the next thing we want to talk about is blood typing. Essentially, when blood is being produced while you're still in your mother's uterus by the liver and the spleen, the formed elements of blood are all basically the same in terms of their function. Okay, the erythrocytes carry oxygen and carbon dioxide, the leukocytes defend us against disease, and the platelets promote clotting. But you can have different blood types, and what the blood types refer to are the presence or the absence of a particular antigen on the surface of a red blood cell. And the two major groups that we concern ourselves with in blood typing in order to avoid what we call a transfusion mismatch reaction, which would result in agglutination and the potential death of the recipient due to a mismatched transfusion, are the ABO group and the RH group, and we'll talk a little bit about why that is. Essentially, when we're talking about the ABO blood groups, we're talking about different collections of sugar molecules on the surfaces of the red blood cells that act as identification tags indicating that those particular formed elements belong inside your body. They are your cells. And so with regard to the different possible blood types in the ABO blood groups, we can either have type A blood, type B blood, type AB blood, or type O blood. Those are the four possible blood types. An individual who has type AB blood is going to have the antigen A and the antigen B both on the surface of their red blood cells. As a result, they will not have antibodies against either one of those antigens, because if they did, what would happen is they would essentially attack their own red blood cells and cause them to lyse, and that would end up now producing an oxygen crisis and a fuel crisis in the body's tissues, and cells will begin to die very rapidly. So in an AB individual, we have both the A and B antigen, but we lack the antibody against the A and B antigen, and for good reason. An individual who has type B blood is going to have the B antigen on the surface of the red blood cell, but they're going to lack the A antigen, and so they'll have antibodies against the A antigen, but not against the B antigen. The result is that if they were to receive blood with A antigen on it, they would agglutinate it and destroy it. So a type B individual can only receive type B blood or type O blood, and we'll get into why type O blood can be used in just a moment. An individual who's type A is going to have the A antigen on the surface of their red blood cells. They will lack the B antigen, and they'll have antibodies against B antigen. So for a type A individual, they cannot receive any blood that has the B antigen on its surface, so they're unable to receive AB or type B blood. They're capable, though, of receiving either type A or type O blood without having an adverse event. A type O individual does not have the A or the B antigen. Instead, they have something called the O antigen, and what's unique about the O antigen is that there's no corresponding antibody to it. So an individual with type O blood can provide red blood cells to any of the other blood types without an adverse event happening, because since there is no anti-O antibody, there's not going to be an agglutination reaction occur in the event that, say, an AB individual, an A individual, or a type B individual were to receive type O blood. However, a type O individual is going to have antibodies against both the A and B antigen. And what that means is that the only blood that they can receive is type O blood. And you might initially think, well, now that's going to be a huge problem, because of the 4 blood groups I can only receive one of the possible 4. But it turns out that type O blood is the most frequent blood type in the United States. So there's always going to be an ample supply of that blood around. Now, you may have been wondering, okay, well, that's all well and good. Why is it that we only have to worry about the recipient's antibodies reacting with the donor's antigens when we're looking at a transfusion scenario, and we don't have to worry about the donor's antibodies and the recipient's antigens? And the reason for that is that when you give blood at a place like Hawksworth, one of the things that they do with whole blood is they remove the plasma, and it's the plasma that contains the antibodies. They resuspend the red blood cells in a saline solution so that they don't rupture. And so when they give that blood as a unit to somebody that needs it, say during surgery, we don't have to concern ourselves with the antibodies that were in the donor's plasma, because the donor's plasma has been removed. So it's not a major concern. What you're looking at here is simply a diagram showing you how it is that we perform blood typing. So what we do is we get anti-sera that we can purify from an animal such as a rabbit that'll contain antibodies against the A or the B antigen, or in some cases we'll also make antibody against another antigen called the D antigen, which is part of what we call the RH group. And what we'll do is we will take a sample of blood from the individual who's going to be transfused, and we'll mix it with the anti-serum. And if the anti-serum recognizes an antigen on the surfaces of the red blood cell of the potential recipient, what will happen is that you'll get what's called an agglutination reaction. And that will result in the clumping of red blood cells, which will eventually lyse. So what you can see here in these panels are examples of what happened when we mix whole blood with the different antisera. So here in the top panel, you're seeing a sample from a patient. One portion of the sample was mixed with anti-A antibody, and one portion of that same sample is mixed with anti-B antibody. And what you see is agglutination in both samples. And so that individual obviously has both the A and B antigen on the surface of their red blood cells. The result is that that individual is type AB blood. Down here you can see agglutination in the B well, but not in the A well. Okay, the sample that's treated with the anti-A antibody does not agglutinate. The sample that's treated with the anti-B antibody does. And so this individual is going to be type B. Down here we can see another patient who shows agglutination when we react with anti-A, no agglutination when we react with anti-B. As a result, this individual has type A blood. And then finally down here, this individual showed no agglutination with either the anti-A or the anti-B antiserum. Result is that this individual is type O. Okay, so that's just a sort of a thumbnail sketch of how we actually perform blood typing. This is just a diagram showing you how it is we generate the antibodies. Essentially what we do is we inject human antigen, either A or B, into the bloodstream of a rabbit. The rabbit will then over time generate antibody against the antigen, and then we'll purify the antibody from the plasma of the rabbit, and that's where we get our antisera from. So in lab, what we did last week was we actually did some blood typing using the blood typing kit, and we were able to, depending on the agglutination reactions in the various wells, determine the blood type of the different samples. So what you're going to want to do is go back and review what we did, okay, and what we're going to do here is take a look at some typical blood typing results so that you can determine the blood type based on what you see in a particular blood typing card or say up on a PowerPoint. So the following slides are photographs of the results of typing for the different ABO groups. On each typing card, anti-A antiserum was added to the blood sample on the left, and anti-B was added to the blood sample on the right. And so what we're trying to discover is what is the blood type of each of these different samples. So what we see here is that we find agglutination in both the A and the B well. The result is that this individual's blood type is AB. Okay, so this individual has antigens against both A and B. They have no antibodies against either the A or B antigen, and the result is what we see in this blood typing card. This individual failed to show agglutination in either the A or the B well. The result is that this individual has type O blood. They're going to have antibodies against both the A and B antigen, and they're not going to have the A or the B antigen on the surface of their red blood cells. They do, however, have the O antigen, but the O antigen does not promote the production of an antibody against it. Okay, it doesn't have what we call sufficient antigenicity to produce an antibody that will attack it. This individual shows agglutination in the A well, no agglutination in the B well. The result is that this individual is type A blood. They're going to have antibodies in their plasma against type B blood. The result is that this person who is type B can only receive either B or O blood. Okay, the next thing we want to talk about are your body's defense mechanisms, how it is that your body keeps you safe from disease. And if we were to look at a typical human being, what we would find is that generally on a day-to-day basis, you're relatively healthy. You're not rife with infection or disease. And believe it or not, that's kind of a miracle, because the environment that you live in is filled with components that are capable of producing disease, called pathogens. Pathogens can either be living or non-living. Some examples of living pathogens include parasites and bacteria, um, and fungi, just to name a few. Examples of non-living pathogens include viruses and toxins, just to name a few. And the fact that we're not overrun with this vast array of pathogens that's in the air that we breathe, in the water we drink, and in the food that we eat is pretty amazing. Your immune system is made up of two types of protective apparatus that keep you disease free. Your innate defense mechanisms are those that use the same type of defense regardless of the identity of the pathogen. So they don't care if the pathogen is a virus or a bacteria or a parasite or what have you. The same method of defense is used in every case. Examples of your nonspecific defenses include your skin, your mucous membranes, and your skin secretions, such as the mucus that you produce, the sweat you generate, even your tears and your urine are examples of secretions that keep us relatively pathogen free. In the event that we penetrate the first line of defense in our innate immunity, there are backups that are present in these tissues that are likely to encounter pathogens that we can think of as our third line of defense, and these include our phagocytic cells, which essentially will eat any incoming pathogen, chop it into little bits, and then display those little bits to our adaptive immunity in an effort to generate now an army of white blood cells that can specifically destroy that pathogen. We also have antimicrobial proteins that are present in our tears and our sweat and our saliva that inhibit bacterial growth. And we have the inflammatory response, which is typical of a wound site that becomes red and painful and swollen and warm. And the idea behind the inflammatory response is to essentially bring blood to the area, and the blood contains white blood cells, which are disease fighters, and that includes the granulocytes and the agranulocytes. And then we've also got these cells that are called natural killer cells that look for any change in a surface antigen on an infected cell, say, or in a pathogen, and what they'll do is produce a basically a chemical reaction. They'll spray the infected cell or the pathogen with chemicals that are contained inside vesicles in a process called degranulation that will promote either the loss of integrity of the cell membrane of the pathogen or the infected cell, or will promote the pathogen or the infected cell to commit suicide, and as a result no longer be a source of disease. Now, in the event that a pathogen gets through these first and second lines of defenses, what we have waiting in the wings are our adaptive immunity, which use a chemical attack tailored to a specific strain of pathogen in order to knock it out without having to produce now a giant army of white cells that use a much less tightly directed chemical attack to wipe out the pathogen. And these include our lymphocytes, our antibodies, and of course the macrophages that assist them. So essentially what happens when we manage to get through these nonspecific defenses is that the macrophages, as well as other cells in the body that are called antigen presenting cells, will engulf the pathogen, will chop it into tiny bits, and will display it on the surface of the cell membrane in combination with a special kind of a protein called a major histocompatibility protein. And what this will do is will serve as a warning sign for the lymphatic system that we've breached now our innate immunity and we have to do something in order to avoid infection. Helper T cells, which are part of the lymphocyte population, will encounter these presented antigens, and you've got 1 billion different types of helper T cells, all with the ability to recognize a different particular antigen. And what will happen is that of those 1 billion different types of helper Ts, there will be a select few that will interact very, very tightly with this antigen that's being presented on the macrophage or the antigen presenting cell surface. And when that happens, what will occur is that these two cells will actually come into contact with each other using the MHC antigen complex as almost like a cell-cell junction. Okay, they'll almost touch, and the antigen presenting cell will stimulate that helper T cell with a chemical signal called an interleukin that will cause it now to divide very rapidly and make many copies of itself. These helper T cells will then, as a result of their ability to specifically recognize that particular antigen, stimulate only other B and T cells that have the same recognition specificity. Okay, so only the B lymphocytes or the T lymphocytes that recognize the exact same molecule are going to be stimulated by this now activated helper T cell. The next thing that will happen is that these stimulated B and T cells will begin to reproduce very rapidly, and they will actually go out and wage a directed chemical attack against the pathogen in order to wipe it out. The B cells use a special protein called an antibody, which is like a magic bullet, which, regardless of where it's produced, will always bind to its target, and when it does, it'll tag it for destruction by macrophages and also by a special series of proteins that's present in the blood plasma called complement. And the T cells that help to defend against disease that interact with the helper T cells are called cytotoxic T cells, and they do something that is functionally similar to what the natural killer cells do, except that they only recognize now infected cells or pathogens that have that particular antigen on their surface. And what they'll do again is produce a chemical attack that'll either cause the membrane to lose its integrity and the cytoplasm to leak out and the cell to die, or again they'll produce a degranulation reaction that will promote the cell that's being targeted to commit suicide, and as a result no longer be a source of the infection. And that's what we call cell-mediated immunity, where the T lymphocyte actually has to show up at the site of the infection in order to destroy the infected cell or the pathogen. And when all three of these are working normally, we tend to stay relatively disease-free. It's when one of these three is compromised that we see an increased incidence of infection and disease, as well as an increased incidence of cancer, because one of the other roles of the immune system is to patrol the body looking for cells that go from being normal to being cancerous, based in large part on the change in the antigens that they display on their surface. So what you're looking at here is just an example of how the nonspecific defenses keep us relatively disease-free on a day-to-day basis. Here we have an example of a puncture wound. What happens here is that the damaged tissue is going to release inflammatory chemicals, in the area around the injury, what will happen is that neutrophils will enter the circulation from the bone marrow, and when they get near the site of the injury, they'll cling to the vessel wall, and then through diapedesis, they will migrate using a process called positive chemotaxis. They'll squeeze through the endothelial cells, and then they'll attack now any pathogen that's in the area, wiping out primarily bacteria, and eventually what will happen is that we will begin to basically close the area off so that the infection can't spread, and then as the pathogen gets wiped out, the next phase will be tissue repair. One of the things that happens in and around a wound or around our nonspecific barriers is that occasionally what will happen is a pathogen will get through, and if they do, they'll encounter a macrophage which will engulf them, fuse this pathogen with a lysosome which will chop it into tiny bits, and then these tiny bits will be disposed of, but portions of them will actually be displayed on the cell surface as a warning to the lymphatic system that something has managed to breach our innate defenses. And again, those lymphocytes that can specifically recognize those foreign particles are going to be the ones that are chosen to reproduce themselves and go out and actually fight the battle for us. Another example of our nonspecific defenses, I told you that there are chemical processes that can compromise membrane integrity. This is an example of how natural killer cells compromise cell membrane integrity in infected cells or in pathogens. What happens is that proteins released from the natural killer cells form a hole that pierces now the membrane of either the pathogen or the infected cell, and that causes the cytoplasm to leak out and the cell to shrivel up and die. So the cell membrane now is completely permeable. The cell can't maintain its integrity, and the result is that the cell essentially just gets destroyed. Okay, it undergoes an osmotic shock, and that's the end of that cell as a source of infection from that point. One of the things that can accelerate this type of natural killer process is the use of antibodies that come again from our adaptive immunity that can attach to antigens on the membrane of the infected cell or the pathogen, and that can initiate now the production of what we call this membrane attack complex that will eventually result in the lysis of the cell and its destruction. So when we talk about our nonspecific defenses, we're talking about a series of physiological responses that are going to, regardless of the identity of the pathogen, eventually result in its destruction. So if we look at the inflammatory response, what we see are the typical changes that happen at a wound site that eventually result in the destruction of pathogens near the wound, eventually leading to tissue repair. We start out with the cells becoming damaged as a result of, say, trauma. We release now chemicals in the area that come from the damaged tissue. Examples include histamine and kinins. What this causes to happen is for blood vessels to dilate. As a result, more blood comes to the area. The area swells up as fluid accumulates, and of course in the blood are white blood cells that are capable of fighting the infection. Okay, the neutrophils, of course, are also attracted by these chemicals, as well as the monocytes. They enter the area, again hitching a ride on the circulatory system, and move in and attempt to A, wipe out the pathogen, and then B, clean up any dead tissue. and then eventually they're going to attract cells that are going to rebuild the damaged area. The increased blood flow into the area results in redness and heat. The increased amount of fluid in the area produces pain and swelling. What happens is that we're going to, as a result, bring more nutrients and oxygen to the area that'll increase the metabolic rate of the white blood cells and also the tissue cells. But at the same time, the increased temperature is bad for the pathogen because it can't deal with the excessive heat. The result is that the pathogen is going to tend to die off, and the white blood cells in your body's tissue cells are going to tend to thrive in this kind of environment. So essentially we're stacking the deck in favor of our own body cells and against those of the invader. The pain and the swelling will result in a limitation of movement of the affected area. This allows time for the affected area to heal. And then clotting will eventually produce a fibrin barrier that will box the pathogen in. The white blood cells can then do battle with the pathogen in this restricted space, knock it out. Very often the results of this battle will be an exudate that we know as pus. So this is the body winning the battle of the infection. And then once the pathogen's wiped out, the fibrin clot is no longer needed. It's dissolved, and then we're able to move on now to tissue repair and healing. Your specific defenses involved the use of really two broad populations of cells: the macrophages, or antigen presenting cells, and then working through the T helper cells, the stimulation of what we call our B cells, which are our agents of humoral immunity, and our T cells, which are our agents of cell-mediated immunity. And essentially what happens when an invader breaches our innate defenses and gets to the point where it finds an antigen presenting cell is it'll become engulfed, it'll be chopped into little bits, and then those little bits will be presented on the surface with a special protein called a major histocompatibility protein, and it's this combination of the protein that comes from the stranger and the protein that comes from our own cell that is going to be recognized by a particular helper T cell amongst that population of 1 billion different helper Ts. There will be a subset of those that will be able to bind very tightly to this presented antigen, and they will be stimulated to divide as a result of their exposure to interleukins, which promote now the rapid cell division of these helper T cells. The result then is that for those cytotoxic Ts and those B cells that also recognize the exact same antigen, they will also be stimulated by this helper T cell, which will produce a chemical that will promote their rapid multiplication. This will generate now an army that will be capable of going out and fighting specifically the invader. And at the same time, one of the interesting things that happens is that as we draft this army of cytotoxic Ts and B cells, we also put aside some of the cells that result from this rapid cell division to use as memory cells, so that the next time that exact same pathogen attempts to access our body through getting through our innate defenses and encountering now our specific defenses, we already have a standing army ready to destroy that pathogen should he show up a second time before he gets a chance to make us sick again. So remember that the cytotoxic T cells work in a manner very similar to the natural killers, except that their method of recognition involves specific antigens on the surface of the infected cell or the pathogen. And the B cells, of course, work by the generation of antibodies, which are going to travel through the blood plasma and eventually bind to the pathogen or the infected cell that contains the antigen that the antibody recognizes, and that will tag it now for destruction. The T helper lymphocytes activate the B lymphocyte cells. The B lymphocyte cells then become plasma cells, which produce massive amounts of antibody, and it's the antibodies that will engage in the attack on the pathogen or the infected cell. This is what we mean by humoral immunity, because the antibodies can act at a distance through the body's fluids, the humors, in order for an effective attack against the pathogen to be generated. And again, without the helper T cell, it would be very difficult for our adaptive immunity to keep us disease-free. This is one of the reasons why the AIDS virus is particularly damaging to the immune system, because it targets the helper T cell and essentially takes it out of commission so that it is incapable of stimulating the Bs and the cytotoxic Ts that would normally go out and fight the infection. And what you're looking at here is just an example of how the different response to an infection occurs the first and the second time that you're exposed to the exact same pathogen. So what happens with the primary response is it takes up to 2 weeks to generate sufficient antibody to really begin to knock out the invader. But the second time that exact same pathogen shows up, the memory cells that have been generated during the primary response produce so much antibody that that pathogen never really gets a chance to produce illness, because essentially they get destroyed before their toxic effects are really they really show up. So you can see here that the second time the pathogen appears, we're able to produce far more antibody in a fraction of the amount of time. Okay, this is a number of weeks here, than we did the first time we were trying to deal with that particular infection. This is why very often, if you were extremely sick as a child, very often you're relatively healthy as an adult. It's because of the preponderance of these memory cells that you've accumulated in the course of fighting all of these different infections. What you're looking at here is just some of the major lymphatic organs of the body. The thymus, of course, sits in front of the heart and behind the sternum and is the site of maturation of the T lymphocytes. That's why they get their name T lymphocytes, from the thymus. Well, the B lymphocytes come from the bone marrow, the red bone marrow, and they travel in the blood to find their new homes, which include lymph nodes, the tonsils, the spleen, and the Peyer's patches, which are found primarily in the submucosa of the digestive system, which is an excellent place to put them, because remember that the food that you eat and the water you drink is not sterile. There are going to be pathogens in it that can potentially get through our innate immunity. So you can see here the tonsils. Okay, you got a set of 5 back here. There's your thymus and your spleen. Of course, your liver is also to some extent a lymphatic organ, and the Peyer's patches, which you find in the submucosa of the gastrointestinal system. You want to make sure that you examine the PowerPoints that touch on the location of the tonsils, know where they are in the sagittal head model, know the location of the thymus, be able to locate the spleen on the torso model. Don't worry about the fetal pig, since we didn't get a chance really to dissect the fetal pig. We're using primarily the models here, so we'll work from those for the purposes of the practical, and you'll want to be able to identify the location of the Peyer's patches, again primarily in the submucosa of the small intestine. Okay, what I want you to stay tuned for is a quick video discussion of how the immune system essentially works, with a brief rundown of the location of the various lymphatic organs. Also a brief discussion on blood typing and blood itself, and stay tuned. The next video podcast, which will be in a separate YouTube link, will discuss the function of the heart. Okay, so I'll see everybody in class.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin, to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups, based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
The lymphatic system drains interstitial fluid from tissue spaces and returns it to the venous circulatory system through lymph capillaries. Lymph capillaries begin as blind end tubes located next to cells and blood capillaries. Once the interstitial fluid enters the lymph capillaries, it is called lymph. The lymph capillaries unite to form lymphatic vessels that have lymph nodes located along them. Lymphatic vessels unite to form lymphatic trunks. These empty into the thoracic duct and right lymphatic duct, which then empty into the right and left subclavian veins. Lymph nodes produce lymphocytes, which help develop immunity to disease. Any foreign substance that enters the lymph node stimulates the immune response and macrophage activity. Circulation of lymph is maintained by normal skeletal muscular activity, movement, breathing, and smooth muscle contraction. Tonsils filter microorganisms from the nose and oral cavity. Spleen filters blood via splenic artery and vein, phagocytizes bacteria, worn-out platelets, and red blood cells. This action releases hemoglobin to be recycled. It also produces lymphocytes and plasma cells. The spleen stores blood and acts as a blood reservoir. Thymus helps develop T lymphocytes. Peyer's-Patch's macrophages prevent bacteria from infecting the intestines.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.